Next metabolism in metabolic regulation is metabolism of glycolipids, and so this is the last topic in metabolic regulation of my portions, and this is a little complex to understand, but it's very quite interesting because it is having major role in membrane and also nerve tissues and also in kidney tissues. So we can have the functions and how it is synthesized and how it is going to be degraded. Okay. So glycolipids are widely distributed in every tissue of the body, particularly in the nervous tissue, such as the brain. Okay, that is main areas are the nervous tissue and the brain. But they occur in the exterior phase of the cell, that is the plasma membrane, and mainly they are involved in the carbohydrate, that is cell surface carbohydrates. The major glycolipid is glucose sphingolipids, and they contain ceramide and one more sugars. And galactosyl ceramide is a major glucose sphingolipid of brain and nervous tissue, found relatively in low amounts of elsewhere. So main thing is the first one is glucose sphingolipids and galactosyl ceramide, and it contains number of characteristics C24 fatty acids, that is cerebronic acid. So galactosyl ceramide, that is galactosyl ceramide, can be converted into sulfo-galactosyl ceramide. It's called a sulfatide. Okay, so the galactosyl ceramide, and it may be converted with the help of sulfur transfer or addition of sulfur, so it is called as sulfatide. So it is also a major component in myelin, and myelin, you know very well, okay, this myelin is a major part in the axons. And the glycosyl ceramide is a predominant simple glycosphingolipid in the extra neural tissues and also occurring in the brain in small amounts. So gangliosides are complex glycosphingolipids derived from glycosyl ceramide that contain, in addition to one or more molecules of sialic acid, that is also involved, and neuraminic acid, that is abbreviated as NAUAC, okay, is the principal sialic acid found in the human tissues. The major sialic acid we are going to deal now, okay, only the neuraminic acid. And we can have one by one, that is the gangliosides are further classified into GM1, GM2, GM3, and GM1 is more complex when compared to GM2 and 3. And you can see here this GM3 is which contains ceramide and one molecule of glucose, and one molecule of galactose, and one molecule of neuraminic acid. Okay, that is what is called as GM3. So how we can name this GM3 means G represents a ganglioside. Okay, that is the main component is ganglioside, and M is a monocyalo containing species. Okay, that is monocyalo species means how many numbers of sialic acid is found, neuraminic acid, and the subscript 3 is the number assigned on the base of chromatographic migration. If you are separating, okay, this one, okay, in the chromatography, and how many numbers, if it is a third band it is called as GM3, and if it is a first band it is called as GM1, and if it is GM2 means, okay, second band. You can see, okay, there is a GM1 is more complex and more molecular weight also, that is why the movement is very less, that is why it is coming in the first band, and second band and third band or GM3 is lightweight, okay, when compared to GM1. So GM1, a more complex ganglioside derived from GM3, and is of considerable biological interest. The major function is it is known to be a receptor in human intestine for cholera-toxin. Only because of this one, the cholera-toxin is going to be bound with this one and creates a lot of complication. Other gangliosides can contain anywhere from 1 to 5 molecules of sialic acid, giving rise to di, tri, sialno gangliosides, etc. Okay, this is the main thing, okay, related to glycolipids, and this is the overall structure of glycosphingolipids. You can see here we can categorize, okay, that one into the neutral glycosphingolipids and acidic sphingolipids. The neutral means so that is ceramide, that is a sphingosine with fatty acid, that is ceramide, and this is linked to one or more sugar residues, that is called as neutral glycosphingolipids. Acidic glycosphingolipids, the same thing, the ceramide, okay, that means ceramide means a sphingosine plus fatty acid, that is linked to one or more sugar residues plus, so one or more sugar residues only here, okay, in the neutral, but in acidic one or more sugar residues plus N-acetyl-neuraminic acid, it is present in the gangliosides, okay, that is GM, and a sulfatide. Sulfatide means a sulfate group is going to be attached. So this is a basic fragment, okay, that is why it is called as neutral glycospingolipids, and acidic sphingolipids a little more complicated. That means along with one or more sugar residues plus N-acetylnuraminic acid going to be attached, that is called as gangliosides, and if the sulfate is going to be attached along with that is called as sulfatides. So this is very simple. The glycosphingolipid degradation is very complex. As I said, okay, maybe this will create a lot of complications in the metabolism. The glycosphingolipids, these are the enzymes involved in the degradation of glycosphingolipids, and finally after the degradation this will be forming sphingosine and sulfate molecule sugars and choline, and all the 4 components are metabolized completely. So this is mainly the lysosomal storage disease, because this degradative enzymes or the degradative enzymes are present in lysosome. That is why the sphingolipidosis, that's called as a lysosomal storage diseases, or the following, or the thing, okay, may be related to this one. So this is inherited enzyme deficiency lead to Tay-Sachs disease, Gaucher, and metachromatic leukodystrophy, and Crab disease, GM1 gangliosidosis, and Sandoff disease, Fabry disease, Farber disease. So all the diseases related to lysosomal storage diseases, and it is very important because all the glycosphingolipids are degraded by the enzymes of lysosomes. If the lysosomes are not able to degrade, okay, this one, and it is all the components will be accumulated within the lysosome, and all the problem will come. Okay, mainly this happens in the neural cells, and the brain tissues are accumulated with complicated fatty glycolipids, and that will lead to abnormal function of the brain and ultimately leads to death also. So this is an example, just you can have a look. So this is a degradation of sphingomyelin and how it is going to be degraded. So this sphingomyelin means sphingomyelinase is an enzyme. Okay, if there is a deficiency of this enzyme, that's called as Niemann-Pick disease. So sphingomyelinase deficiency is a major issue, and enlarged liver and the spleen filled with a lipid. Severe mental retardation, and especially in the children, and neurodegeneration will happen. Death in early childhood, that is that will happen. The persons affected with this enzyme deficiency, they will not survive. Maximum possible, okay, they may survive, okay, 3 to 6 months of time, and within the duration, okay, they will get a lot of complication like mental retardation, neurodegeneration, and enlarged liver, and atrophy of the liver, and finally death may happen. So you can see here, okay, this is the microscopic image of lipids seen in spleen cells from a patient with the Niemann-Pick disease, and the foamy appearing cells containing sphingomyelin. You can see here, okay, these are all the spaces found in the tissues of spleen, and this is accumulated with the sphingomyelin. The sphingomyelin is not degradable because this enzyme is not present. Due to that, this okay, this ceramide, okay, this is sphingomyelin is accumulated, okay, within the cells. So coming to the synthesis of sphingomyelin, how the synthesis has happened, what is the source of synthesis? We can start with palmitoyl-CoA, that is a fatty acid, and along with the combination of serine molecule, okay, this will synthesize spinganine, and the spinganine is converted into sphingosine, okay, by FAD to FADH2 conversion, and the coenzyme is liberated, and FAD to FADH2, okay, gets, okay, 2 hydrogen molecule, okay, from here, and the fatty acyl moiety of the fatty acyl CoA donated to here. So this will be linked with spinganine. The spinganine is converted into sphingosine. So this is the sphingosine moiety along with the fatty acids called a ceramide. So this is sphingosine. Spinganine is converted into sphingosine with fatty acyl moiety from fatty acyl CoA, and this is called a ceramide. So this is the structure of ceramide, and by phosphatidylcholine, that is a donor of choline molecule, the choline is going to be attached with the phosphate, and so this one, that is CH2O, okay, this portion is going to be attached with the choline, and this is called as sphingomyelin. So this sphingomyelin is very, very important for the neural and also the neural cell function, and all the functions of, okay, including the spleen and the liver cells also. So coming to the classification of the structures, we can classify, I already told, okay, this I already classified, okay, this one. the glycosphingolipid is converted neutral sphingolipids and acidic glycosphingolipids. And you have to think about, okay, this one neutral glycosphingolipids contains ceramide. So ceramide, I told, okay, this is ceramide. So this is ceramide, and so ceramide linked to one or more sugar residues. Sugar residues going to be attached. So that is the main reason, okay, that is why the sphingomyelin is going to be synthesized. Okay, next one is the neutral sphingolipids. The further classification examples are cerebroside, okay, that is ceramide plus glucose. CER means ceramide. You can see here ceramide means CER, GLC means glucose, gal means galactose, and galNAc means N-acetyl galactosamine. So globoside and globoside, okay, this is the another version of globoside, okay, that is the phosphine antigen, and this is more complicated than the globoside of lactosyl ceramide. And this is neutral glycosphingolipids. Next one is acidic sphingolipids. I already explained about this one in here. Acidic glycosphingolipids are ceramide which combines, okay, the lipid composition, okay, that is called as sphingosine plus fatty acid, and along with that one or more sugar residues, that is which is similar to this one neutral sphingolipid. Along with that N-acetyluramic acid going to be attached, okay, that is why it is called as gangliosides and sulfatides. So we have in this category only two types, gangliosides and sulfatides. So we can see here gangliosides. So number one is ganglioside and sulfatide. So gangliosides are the most complex glycosphingolipids and found primarily in the ganglion cells of central nervous system. And the G means, okay, the ganglioside, and nana means N-acetyluramic acid, okay, this one. And why it is called as nana means, okay, this nana is very, very important. That means this is involved in the the function of GM2. So without N-acetyluramic acid, there is no function of GM, and this and contain one or more molecules of nana. Without nana, so there is no G, so that is why nana is very important. Nana means nothing but N-acetyluramic acid. So N-acetyl galactosamine attached with galactose and glucose, and with the ceramide and N-acetyluramic acid, okay, going to be formed. And this N-acetyluramic acid is the major component, and within this the GM2, and GM2 comprised of N-acetyl galactosamine and ceramide and glucose, galactose, and nana. So GM2 means that is a type 2. Okay, the GM1 is the first one in the chromatographic separation, GM2 is the second one, and GM3 is the third one. GM1 is more complex molecular weight and a very highly complex one when compared to GM2 and GM3. And this is given in further diagrams, okay, I'll discuss. And the next one is sulfatides. Glycosphingolipids, that's called as sulfatides or cerebrosides, that contain sulfated glycosyl residues, that is sulfur moiety is going to be attached, and this is very much predominantly found in nerve tissue and kidney. The major functions are in kidney also. And synthesis and degradation, how it is synthesized and degradation, we are going to see that one. The enzymes involved in the synthesis, usually the glycosyl moiety transfer by glycosyl transferases, and sulfate moiety transfer by sulfotransferases. You know that, okay, sulfate is not easily transferred like other molecules. For example, this carbon donor is S-adenosyl methionine, and at the same time sulfur donor is PAPS. So you can see here, this is a sulfur moiety, so this is donated by a molecule, this is active molecule PAPS. PAPS means 3-phosphoadenosine 5' phosphosulfate. This is a donor and donates this one, that means sulfur. So the PAPS is also the sulfur donor in glycosaminoglycan synthesis and steroid hormone metabolism also. We already studied the steroid hormone catabolism, and this is very important, the PAPS one. So coming to the Gaucher disease, you can see here the bone marrow cells from the patient with Gaucher disease. The crumbled tissue paper appearance of the Gaucher cells, and this is caused by glucocerebroside accumulation due to the elongated lysosomes filled with the glucocerebroside, the deficiency. So the degradation of glycosphingolipids, and this will cause, okay, this one, the disease, and sphingolipidosis, Sphingolipidosis means there is accumulation of sphingolipid, okay, within the cells, okay, mainly due to the lysosomal hydrolase deficiency, and this will create a lot of complication, and this may lead to early death also. You can see here, this is the overall sphingolipid synthesis. Ceramide converted to sphingomyelin by conversion of phosphatidylcholine to diacylglycerol. The choline is going to be attached with the ceramide, that is why it is called as sphingomyelin. The ceramide again, okay, by the use of UDP galactose converted to UDP, and the galactose might be going to be attached. That is why this ceramide plus galactose called as galactose cerebroside. And due to the PAPS, the PAPS means donor of sulfur, okay, just now I told, and that will donate one sulfate moiety, okay, to this one, and this one is converted to sulfatide. And ceramide, in the presence of UDP glucose, the donor is UDP glucose, and glucose is donated and UDP is liberated. So that is why this ceramide, so similarly to this, the galactose cerebroside and the glucose cerebroside will be formed. And 2 or more UDP sugars will be involved here, that is globoside will be formed. From globoside, ganglioside will be formed. So ganglioside means, okay, the CMP plus NANA, and CMP is liberated and NANA going to be attached, okay, that is why it is called as ganglioside, that is simply called as GM. So I already shown this, the composition of GM, you can see here. So this is the NANA one. So NANA is coming from CMP NANA, and NANA is going to be attached with the galactose. So this is the overall overview of sphingolipid synthesis. And this is ultimate the degradation of sphingolipids in the lysosomal enzymes affected with the different genetic diseases also. And all the diseases are autosomal recessive, and I think except one disease, Fabry's, okay, Fabry's disease, and which is S-linked, and all the other can be fatal, okay, in the early life. The patient will not survive, unfortunately, because due to the mental retardation and abnormal liver function and kidney function, and this may lead to abnormality in the normal metabolism and leads to death. I will elongate, okay, this one. Can see here, this is GM1. So GM1 means the galactose and N-acetyl neurominic acid, and along with galactose, glucose and cerebroside, and attached with NANA. Okay, this is called as GM1. That is the most complicated one, that is GM1. So this gangliosidosis, the first one, GM1 gangliosidosis, accumulation of GM1, accumulation mainly in the neurological tissues, and sometimes, okay, in the spleen also, and sometimes in spleen and liver. This may lead to cherry red macula and skeletal abnormality or deformities, and hepatosplenomegaly and neurologic deterioration, and also accumulation of creatine sulfate also. So this is the deficiency, that is beta galactosidase deficiency. If the beta galactosidase is present, automatically this one is degraded, the galactose is removed, and if the galactose is removed, automatically, okay, this is the GM2 will be formed. If the GM2 is if not degraded by beta hexosaminidase, okay, if this enzyme is deficient, that is called as Tay-Sachs disease, and accumulation of ganglioside GM2, okay, rapid and progressive and fatal neurodegeneration will happen, and sometimes leads to blindness, cherry red macula, muscular weakness, and seizures will be formed. And this one involved in the removal of, you can see here, this galactose with the neurominic acid, and the galactose with the neurominic acid will be removed, okay, this one, and remaining thing is, okay, this fragment, NANA with galactose, glucose and ceramic acid. Okay, this involved in the accumulation, that is, this is not maybe the very familiar one, so there is no pathology related to this one. That is why GM3 not involved with any pathology, because neuramidase enzyme is very common, and neuramidase removes any, that is, NANA is going to be removed, and if the NANA is removed, the remaining part is there, the lactosyl ceramide, and this lactosyl ceramide involved with the beta galactosidase enzyme, the galactose is removed, and glucose with the ceramide is the remaining fragment, and beta glucosidase. So this is the enzyme will cleave the bond between the glucose and the ceramide. So if the glucose is not liberated from glucose with ceramide, okay, this will create a complication, okay, that is called as Gaucher disease. The Gaucher disease is nothing but accumulation of glucose cerebrosides because glucose cerebroside is accumulated, and most common lysosomal storage disease and hepatosplenomegaly, osteoporosis and long bones, and seen a central nervous system involvement in rare and infantile and juvenile forms. So next one is if this is working, maybe the beta glucosidase is present, automatically this one is glucose liberated and ceramide will be there, and ceramide is further degraded by an enzyme ceramidose. So this the deficiency involved with the enzyme, okay, that's called as the Faber disease, and accumulation of ceramide, painful and progressive joint deformity, and subcutaneous nodules and lipid laden cells, and host cry, and the tissue, so the granulomas also. So this will liberate fatty acid. If it is working, the fatty acids are liberated and sphingosine will be there, and the sphingosine is further metabolized. So that is one. Next, in the branching, maybe we can come to the top one, the globoside. We have to think about this also. The globoside is this is a composition of globoside, and the globoside is degraded by beta hexaminase A and B. So if this enzyme is that is deficient, that will create a disease called as Sandoff disease, accumulation of this GM2 and globosides, and same neurological symptoms, okay, like Tay-Sachs disease, the same, okay, this one and this one, the symptoms are same, and sometimes, okay, the differentiation is also a big problem. Only we can get the biopsy tissues for the further separation of GM, and based on the electrophoretic separation we can identify, okay, which one is accumulated, either GM2 or otherwise globoside. This is mainly the globoside accumulation, and this one is GM2 accumulation. So these two having the different molecular weight, so we can identify easily by the electrophoretic separation from the biopsy tissue. So this one involved in the degradation of this one, the galactose with neuraminic acid is going to be liberated, and remaining this fragment is here, and that is alpha galactosidase enzyme is involved to remove galactose, and remaining fragment is galactose, glucose and ceramide. So this galactose is liberated by alpha galactosidase. The deficiency of this one leads to Fabry disease and accumulation of globosides, and reddish purple skin rash will be formed, and kidney and heart failure, okay, will be happening. So next branching is here, okay, that means ceramide synthesis. That means the phosphorylcholine ceramide, the sphingomyelin to ceramide synthesis. Okay, this one, the further sphingomyelin is degraded by the sphingomyelinase enzyme, and Niemann-Pick disease will be formed, either the type A and type B, and accumulation of sphingomyelin, especially in the spleen, that leads to hepato or that is splenomegaly, and this leads to cherry red macula. And further, okay, this side, okay, that is sulfatide, the removal of sulfate, okay, mainly the enzyme, okay, sulfatase, aryl sulfatase, and if it is deficient, that is called as metachromatic leukodystrophy. The accumulation of sulfatides will be there, and cognitive deterioration, this mentally retardation will be there, and progressive paralysis and dementia, and there is no possibility of survival, okay, here, and this is more pathetic disease. And next one is the galactose with the ceramide, liberated by the galactose is liberated by beta galactosidase enzyme, and this beta galactosidase enzyme deficiency leads to Crab disease. This is also more pathetic, mental and motor deterioration, and ultimately leads to blindness and deafness, and mostly the person will die within 3 to 4 months of duration. And these are all the things, okay, about the complete degradation of GM and globoside. So this is the one I already explained. This is galactose one, attached with the sphingosine with the fatty acid, and the total it is called as ceramide. And ceramide attached with the glucose and galactose and neuraminic acid, N-acetyl galactosamine with the galactose. Okay, this is called as GM1, the highly complex one. So with this we can conclude glycolipids. Thank you.